Okay, question 7. Industrial processes can use immobilized enzymes. Give one reason why using immobilized enzymes can cost less money than using enzymes that are not immobilized. So this is basically comparing immobilized enzymes to free enzymes. So these would be examples of immobilized enzymes. We would put them in usually alginate beads. And you can imagine one of the advantages of using immobilized enzymes is so that you can reuse the enzymes. So enzymes are reusable. Because imagine if you had like a beaker, right? You have your substrate and then you added like the enzyme into the substrate and then you would then form the product. So now you have the product and the enzyme in the beaker together. How are you going to separate out the enzyme and the product? When we use immobilized enzymes, we don't have to worry about separating them out. Because because of that, we're able to reuse it. Okay, so the next part, the diagram shows the apparatus used to produce glucose and fructose using immobilized enzymes attached to alginate beads. This is substance X, and then it gets broken down to glucose and fructose. Name substance X, that would be sucrose. So you do need to remember this. Sucrose gets broken down to form glucose and fructose. All right, let's do the third part. So I've included a little diagram here. This is the closest diagram that I could find. So just ignore the cell suspension part. So here, this is how you go about preparing alginate beads. So number one, you would have the sodium alginate, and you would make a sodium alginate solution. So to do that, you would mix sodium alginate with distilled water. And you'd mix them together until it dissolves. Then, using a syringe, you would add drops of that sodium alginate solution into calcium chloride solution. So you add it to the calcium chloride and that would harden it. And then once you get the beads, you would then wash them. Okay, let me just make this one smaller and write this one out. So first, mix sodium and distilled water until the alginate has dissolved. Then using a syringe, fit to calcium chloride. After that, wash the beads. Okay. Let's move on to part B. Which process is involved in the conversion of glucose to glycogen? So glucose is a monosaccharide, glycogen is a polysaccharide. All right, so let's look at each of the statements one by one. Glucagon is released from the pancreas and travels to the liver. Glucagon is a hormone that is released from the pancreas. However, it causes glycogen to be converted to glucose. So therefore it's not A. Let's put it's not A. All right, let's look at statement B. Insulin is released from the pancreas and travels to the liver. That is correct. So the correct answer for this is B, because it is insulin that is responsible for converting the glucose to glycogen. Okay, part C1. The total population of the United Kingdom is 66.65 million. Diabetes affects 6% of this population. Calculate the number of people with diabetes in the United Kingdom. So we would just want to find 6% of this number here. So 6% is the same as 0.06. Right, so we would do 66.65 million. And you can see here in the answer, they've written million here. So you don't need to add all of the zeros for million, just write 66.65 and then times that by 0.06. Let me just calculate that. So 66.65 times 0.06, that would be 39.9. That would be 3.999, which is the same as 4. So that would be 4 million. All right, the last part of this question. Surveys were carried out in different areas of the United Kingdom. These surveys collected data on the number of people in each area with diabetes. The information collected is shown in the box. So determine the median number. So median, in order for us to find what the median value is, you want to be able to order these numbers from smallest to largest. So let's go and do that. So the smallest number here we see is 38. So I'm just going to cross that. No, I won't cross that. I'll just underline it once I've written it down. The next number here is 49. The next number here is 51. So we need to do this in order to find the median, right? The next number is 62. Next number is 65. Next number is 78. Then we've got 82. Then 91. And the last number, 96. So let's look at the number in the middle. There we go. So the median is 65. Question 8. Discuss the advantages and disadvantages of the use of embryonic and adult stem cell in medical research. So if you recall, stem cells are basically cells that are able to undergo mitosis and they don't differentiate yet, or they haven't specialized into cells yet. So there are two stem cells that we use in medical research, embryonic stem cells. Let me just switch this to a pen. So they originate from embryos, right? So they're found in the early stage of development, whereas the adult stem cells are found in adult tissue. So for example, our bone marrow. So our bone marrow consists of stem cells that can differentiate to form red blood cells and white blood cells. Because this question is worth 6 marks, we would need to split this answer into 2. So this section here is worth 3 marks, and this section here would be worth 3 marks. But let's play a little safe. Let's do 2 pros and 2 cons for the first section and 2 pros and 2 cons for the next section. So one of the advantages of embryonic stem cells is they can differentiate to any cell in our body. So the advantages is that can differentiate This is why embryonic stem cells are really good for research and also for treating diseases like Parkinson's diseases. So you can have these embryonic stem cells differentiate into neurons, for example. This is why they are used to treat Parkinson's. So that's 1 mark here and another mark there. So those are the 2 pros. Let's talk about the cons. So where are we getting these embryonic stem cells from? We're actually getting it from fertility clinics. So in fertility clinics, let's say a couple walks in and they undergo IVF, in which you would fertilize the egg in the lab and then implant the embryo to the mother, or inside of the mother. And sometimes they're embryos that get discarded, and for some people, it might be against their belief to do that because they might see embryos as potential life, so it would be considered unethical or even murder. So let's write that as one of the disadvantages. Are that may view this as unethical. Okay, another potential risk of using embryonic stem cells is that they may cause cancer. May also cause cancer as well. All right, let's move on to the next part. Okay, what's really cool about adult stem cells is that if you were to take adult stem cells from the patient, they are less likely to get rejected by the immune system because the immune system doesn't see it as foreign. So that's actually an advantage there. Using adult if from the patient, the patient will not experience rejection. Ooh. Rejection. So rejection is when the immune system sees something as foreign and rejects it. So usually it could be organ donation or stem cell transplantation. Okay, so this is one mark, and where's the other mark here? Oh, originated from the patient or taken from the patient. Sorry, I should underline this one as well. This is another mark. So that's 1, 2, 3 marks there. Another mark there, that's 4. 2 marks here. Let's talk about the disadvantages. So a major disadvantage of adult stem cells is that they can only differentiate into a smaller range of cells. So let me write that down. The advantage can only differentiate into a limited range of cells, I should say. Oh, I think it's lagging a little bit. Another disadvantage that you can also say is that there's a very small number of these adult stem cells found in our body anyways. So let me just write that down. There is also a small number of adult in our body. Alright, so limited range of cells, so differentiate into limited range of cells, that's another mark. And small number of adult stem cells in our body, that's the 4th mark. Alright. Okay, we're finally in the last question. So question 9. In England, 31% of men and 26% of women have high blood pressure. Explain how medication can be used to treat people with high blood pressure. Okay, so this is one of the medications that you need to know, and that is called the ACE inhibitors. So let's go over that together. So just a little bit of background here. So when your kidneys detect low blood pressure, it actually secretes an enzyme called renin into the blood. And that causes this protein angiotensinogen to get converted to angiotensin. And then ultimately angiotensin would cause the constriction of the blood vessels and a rise in blood pressure. So this medication called ACE inhibitors inhibits this enzyme over here. So if you inhibit this enzyme, you would have less of your angiotensin, and therefore you would have less constriction of blood vessels and less of that rise in blood pressure. All right, so let's include that in the answer. Just going to make this smaller. Okay. Where's the pen? Oops. Okay. ACE inhibitors used to inhibit the enzyme responsible for creating angiotensin. There is less angiotensin. The blood vessels constrict less. And that would decrease the blood pressure. All right, so this is the marks that you need to hit. ACE inhibitors, responsible for angiotensin, inhibits the enzyme that is responsible for creating angiotensin. When there's less angiotensin, when there's less angiotensin, the blood vessels constrict less. So these are the 4 marks. There's also another method in the marking scheme that you can add. You can say that another way to treat people with high blood pressure is to use beta blockers because that prevents the release of adrenaline, which would then cause the heart rate to decrease. Okay, so let's move on to B1 and B2. So I've included this diagram over here. Let me just make this larger. So here we have the blood pressure changes in the different blood vessels. So here, as you can see here, with your aorta and your arteries, there is a drastic change in blood pressure. You can see it goes up and down. So the reason why the blood pressure goes up is because the ventricles are contracting. When the ventricles are contracting, it pushes blood out of the heart, and that's why you have these peaks that you see here. And we refer to them as systolic pressure, right? And when our ventricles are relaxing, it causes the drop in pressure, so that would be diastolic pressure. So let's write that down in the answers. Let's write them down here. Okay, describe what is meant by systolic blood pressure. So systolic pressure, okay. Systolic pressure refers to the pressure of blood exerting against the walls of the vessels. Walls of the, I should say, artery vessels. And to the walls of the artery, sorry, against the walls of the artery when the ventricles are contracting. Ventricles are contracting. So this is one mark. This is another mark. And this is another mark. All right, diastolic pressure is the pressure in the arteries when the heart is, or when the ventricles are relaxed. So this to the pressure of the arteries when the ventricles are relaxed. Okay, this is one mark, and this is the other mark. All right, and I think there's only 2 marks for this. Let me just check. Yep, there's only 2 marks for this. All right, and that's the end of this paper.